So this is an example of a hole-mounted worm that's been placed on a slide. This is what it would look like if it was embedded in the gut. My name is Emma Martinez, and I'm a senior at Rockhurst studying biology and philosophy. And I've been doing research with Dr. Cielocha for about 3 years now. We've been looking at a previously undescribed species of tapeworm collected from the gut of a ray called Urogymnus asperimus 1.
For the last 2 years, I've had students working on the description of this new species, and also I've been working on characterizing the sperm ultrastructure and learning how the tapeworm attaches to the host's gut.
This species of tapeworm is very unique morphologically. It has an apical organ called a scolex, and it's kind of glandular and muscular by nature, and it attaches very intimately to this host tissue. So we basically have taken the spiral valve out, which is the gut of the ray, and we've kind of looked at this interaction between the host and the parasite. This is the scolex, and that's that really intimate attachment site between the tapeworm and the host. So we're just trying to understand how deep it's embedded, if there's any response elicited, and just kind of that interaction between the host and the parasite. These are really undescribed species, and there's really no histological staining or any research that's been done on these worms in particular because they're pretty new.
This project in particular, Emma has been working on for a couple of years. She actually started working on the spermatozoon project, and that is just a series of images that we're analyzing that were taken with a transmission electron microscope. And we're trying to reconstruct the three-dimensional anatomy of those sperm from this tapeworm. For this project, in terms of how the tapeworm attaches, she's been doing a lot of histology, so sectioning that tapeworm and learning about how it attaches to the host epithelium. And we're going to start doing some specialized staining.
This tapeworm has been embedded in wax previously, and so we're using this microtome knife. It's very sharp. There's a really super precise blade. And so basically what we do is we take sections of this embedded specimen until it makes like a ribbon, and then essentially we will transfer this ribbon with the specimen present. It's a very, very, very thin section of wax, and then we will end up placing it upon the slides. Our plan for this next semester is to do 2 rounds of staining, and the first stain will be a periodic acid shift staining with a hematoxylin counter stain. This periodic acid shift, or PAS staining, will show if there's any white blood cells present from the host. So again, we're looking at that specific interaction between the host and the parasite, and then we will also be staining them with a trichrome green stain. And so that will basically tell us this characterization of tissue responses. So like if there's fibrous or collagenous tissue present, that might show us that there's some sort of ulcerative or tumor-like response from the host. So we really are kind of trying to characterize the overall immune response at this host-parasite interface.